Okay. Hi, everyone. Thank you for coming to my talk. I hope you enjoy this conference and learning, because learning and memory are the topics of my talk as well. So I hope you guys are really going to be enjoying that. And it's not only learning and memory, but most importantly, how do psychedelic drugs, which is the topic that I guess we're all interested in, affect learning and memory. So I'm just going to briefly introduce myself for those of you who don't know me yet. So my name is Maria Balaet, that's how you pronounce it. And I'm a neuroscientist working at Imperial College London in the Computational Cognitive and Neuroimaging Laboratory. It's not in that building over there, but that's the prettiest that we've got at Imperial. And yeah, I basically became really interested in learning and memory back when I was a student at the University of Birmingham. And much of my work there, research work, has been focusing on memory modulation in models of PTSD and addiction using pharmacological agents, using a paradigm that I'm actually going to describe today. And I found it really interesting considering that us as humans have a lot of memory in our life, but we don't seem to be able to forget things very easily. And what we can remember, I thought, pretty much makes up what we're dealing with on a daily basis. So I started to look at memory as a way of describing consciousness, really, because this seemed to me like the most plausible way that we could be explaining what we are doing on a daily basis. And since we're talking about consciousness, let's delve a bit into evolution as well, because consciousness is unique to humans to some extent, unique to us as we know it, although it has been arguably present in other species. And I guess you're familiar with these guys portrayed over here, one of them being Charles Darwin and the other Alfred Wallace. So they are the guys responsible for the theory of evolution. But also they had some interesting chats and insights about consciousness. And this guy, Alfred Wallace, kind of disagreed with his friend Darwin afterwards, and he published a paper trying to state that consciousness is not something to do with biology necessarily, but there has to be some greater mystery about it that science could not theoretically explain. And I'll let that to you to decide, but for the moment I'm going to portray a bit of a different explanation than that. This guy here, do you guys know who this guy is? Just, should I introduce him? Okay, so this guy here, I thought it's a really, really cool guy. His name is Gerald Edelman, and he's actually a Nobel Prize winner. Not for anything to do with neuroscience, but with immunology. And that's not what we're going to talk about today, no. What's interesting about him is that he actually came up with a theory of consciousness and published this short book. It's only 30 pages long, and you can find a PDF online at the simplest Google. It's called Wider Than the Sky, and there he describes this theory of consciousness that he coined as neural Darwinism. So it has to do with evolution as well. So in his view, he basically states that consciousness is a product of the brain as a biological mechanism, and that it has a couple of characteristics that we can look for. But what is actually important about the fact that consciousness, in his view, is a product of the brain and the brain only, so product of evolution, is the fact that this makes it accessible to scientists for us to measure it. Because just as some of my colleagues like to say, if you can't measure it, it doesn't exist. Well, as a personal story on the matter, I had a chat with a psychologist the other day who was trying to say that, oh, but we can't measure hope, for example. Well, maybe not. We don't really seem to have very robust measures for such cognitive processes yet. To come back to Gerald Edelman, characteristics of the consciousness. He first said that the consciousness has to be embodied. So it has to be in a body, basically. No one has seen a free-floating conscious spirit around, I hope. But if you do, put it in this box so we can study it. That box. it has to be a multi-dimensional box, so, you know. It's also personal, so each individual experience of consciousness is highly unique to the individual, and maybe that's what makes it so difficult to study and so difficult to look at. It's also continuous. You don't find yourself, I mean, I hope you don't find yourself like every single moment going from one conscious state to another, or like from consciousness to non-consciousness. That would be cool. And it's also dependent on attention, because obviously you can't focus on everything around, but depending on what you're focusing on at a particular moment, that gives your own experience. And to kind of map the whole concept of how we're going to describe consciousness, at least for this presentation, let's just take the present moment and think about you guys being here in a room. You're paying attention, I suppose, and you're also aware of your flight time tomorrow. You're also aware of what awaits for you at home. You're aware of a lot of things that you probably learned in your practices and everywhere on a daily basis. You remember you had lunch, and so on. And this is basically the perfect explanation that frames what Gerald Edelman was saying, that consciousness is the remembered present. And as I was saying earlier, it's memory in action, really. But how do psychedelics influence that? So I'm just going to take the example of LSD to briefly introduce psychedelics, because I'm quite sure that all of us are familiar with what they're doing at this point. So this is the LSD molecule in a very pretty picture that I took of Google Images. And psychedelics are well known for causing these profound effects on the human body and on the consciousness. They basically change our perception of the self, our perception of reality. They can cause things like synesthesia. They enhance the visual field and all sorts of things that have not even been described before. Their potential seems to be endless. But the way that these compounds work, I'm just going to briefly touch on that, again with LSD, as described by the researchers at Imperial College London in the Psychedelics Research Group, my colleagues from the 5th floor. They do that by decreasing the blood flow activity in something called the default mode network, thus allowing the other regions to communicate with each other. And I'm just going to briefly explain this. I'm not sure how many of you are clinical neuroimaging people or so on, but there was this big newspaper in the UK a couple of years ago who published this title that LSD causes a lot of orange in the brain. This is not orange. This is blood flow. And these are the slices that are here. So I think it's really cool to actually know how to interpret a scan, because I'm very well aware that not all of us are working in this kind of environment. And the way that this happens is really at a molecular level. So psychedelics act by being agonists at the 5-HT2A receptor, which is this one over here. So do you guys want to get an explanation for this? Yes. Yes. Okay. So basically, when psychedelics act on the brain, they act on this 5-HT2A receptor, and then they trigger this internal protein cascade that pretty much makes the effects manifest. And what's important about this is this GQ protein. So GQ protein has two subunits. One is called the alpha subunit, and one is called the beta subunit. And this is the alpha subunit here. They are usually together, but when something acts on them, they dissociate. So this one stays here, and this one triggers this, which is called the phospholysis C, to trigger the release of inositol phosphate and diacylglycerol. And then, Yes, yeah, sorry, this is inside the neuron, obviously. Like the receptors are on the membrane of the neuron, so the membrane is the surrounding part of the neuron, and then they trigger this calcium activity, and so the psychedelic effects that I just described happen. And honestly, like, I think this is where the magic lies. Like, if we can explain how this happens, why it happens, most importantly, maybe we can actually find some answers about what's really triggering our curiosity about these drugs. So yeah, that's basically what leads to this. Coming back to memory now. To introduce memory a bit. Oh, you can't see that. I was sure this was going to happen. So the easiest way, in my opinion, to define memory is all the sensory information from outside the body being processed by the brain and resulting in something that we can remember, being memory. And just before we move on to the other fun stuff, does anyone else have a different definition of memory that would like to share? I'd be really interested in that. Yeah? No? No? No one has a different definition of memory. Okay, amazing. No, honestly, like, I'm a big fan of like debating definitions with people. So, memory. Memory has some processes associated with it. We can basically break it down in terms of what happens. So we have the encoding stage, which is basically taking this information, processing it, and keeping it in the brain in a form of another. And then comes the consolidation process, which means that we take that information, the sensory information from the outside, and consolidate so we can keep it in this storage space. You can think about it like a library, for example. Then we can access that by retrieval. And here I put in brackets my actually favorite part, which is the very basis of neural plasticity, in my opinion at least, and in some other scientists' opinion, which is the reconsolidation of memory paradigm. And I'm going to touch a bit more about this later, but what it basically states is that memories can be edited and modified upon retrieval. So what I'm going to do now is I'm going to take each of these processes in turn, describe what they are, and then talk a bit about what psychedelics are doing to influence each and one of these processes. So let's start with encoding. Obviously, there are more types of stimuli that can come up from the outside, but these are the ones that I decided to put up on the slide. So we have visual encoding, acoustic encoding, and tactile and semantic encoding that seem to be predominantly the stimuli that the brain seems to remember. And all of this information coming from the outside gets processed in the brain, as I just said. But here's a very good moment to actually say what psychedelics might do. In the current baseline consciousnesses now, we look around, we obviously have all this visual information that we can encode in our brains, but on a psychedelic drug, the visual field, it's not that visual field enlarges or increases, but it definitely changes. And it's also the fact that we may or may not have higher perception of the acoustics around. Therefore, this is another stimuli that can be amplified by the use of psychedelics. I'm not going to talk about the tactile one necessarily, but like the semantic one, I find to be one of the most important to touch on, because semantic encoding refers to the encoding of meaning. And what it states is the fact that the more meaningful information is to you, the more easily it would be recorded in the memory and it would be stored there. And as you've probably heard in the previous talks, psychedelics tend to increase the meaning of a certain situation. They definitely can be regarded as tools for finding meaning, creating meaning, where there would previously not be any. And a good example is, anecdotally, there has been this story circulating on the internet about this guy that found the meaning of life by looking at a Coca-Cola bottle. So meaning can be found much easier with psychedelics. I told you it's going to be fun. The region, I'm going to touch a bit on the regions of the brain that are involved in this encoding as well. So it's the thalamus especially that provides like this hub of sensory integration for all the stimulus coming from the outside. The only stimuli from the outside that wouldn't go through the thalamus is the olfactory stimuli. So basically smelling, that doesn't go through the thalamus. It has a different pathway, but we're not talking about that now, hence why it's not there. And it's also the prefrontal cortex, which is very important in decision-making, in choosing what you want to remember or not. The cerebellum, that's important for procedural memory encoding. So procedural memory is like riding a bike, for example. Many other things, like juggling maybe could be regarded as a procedural memory. And the amygdala, it's also really important because the amygdala is the hub of basically emotional regulation. And a good example here would be with fear memory. If you have a strong feeling of fear against something, you're probably going to remember to avoid that thing in the future, and that's a very fundamental evolutionary adaptation that we have. Then it's the hippocampus, and that I'm pretty sure it's widely known that it's really important for memory, because it has been shown by lesion studies that if you remove the hippocampus, then you might suffer with retrograde amnesia, and that basically means that you can remember everything up until the point of hippocampal removal, but you can't form new memories. And just to support this theory, actually, there has been some, it wasn't with psychedelics, but it has been this really, really cool experiment at Cambridge University recently. They showed sequences of a film called Forrest Gump, you might have seen it, to people in the scanner, and they asked them to try to remember each sequence in turn. And what they noticed is that the activity in the hippocampus increases as they're trying to remember, and just as the sequence ends, it decreases. So basically, we could say that the hippocampus is literally just like this region that allows us to encode memories, compare them with previous ones, and make new associations. And I just tried to find something about whether psychedelics have some direct influence on the hippocampus, but the literature is not very clear on that. However, there are a couple of other things where the literature is much clearer, and I'm going to touch that in the upcoming slides. But just before I do that, I just want to mention the concept of an engram. And engram is how memories have been regarded before as being stored in the brain. So this is like a hypothetical biophysical or biochemical change in the neurons that represents a memory. But honestly, like, no one ever found evidence for an engram being in the brain, so there's not a particular bit that you can extract and delete that memory. So what do? We need to find a way to induce forgetting in the people, right? For that, moving on to consolidation for a bit. So consolidation can be of two kinds, synaptic consolidation and system consolidation. And just to briefly explain synaptic consolidation, which is a really fun process, I promise you it's fun. So you see this magnesium that's over here. So this usually blocks what is called the NMDA receptors, and these are the most important receptors in memory. If you want to go out and read a research paper and it mentions this, remember it's really important in memory. When a cell gets depolarized meaning that there's some electrical activity going into it, then this magnesium gets taken out and glutamate is released in the synapse. This is a synapse. And then this triggers calcium influx into the neuron, and then that's basically the whole process of how a memory gets consolidated. And here is where I think that psychedelics play a really key role, because there are studies that do agree on the fact that they do cause increased glutamate extrasynaptically, and they do definitely cause a lot of activation of the brain and brain stimulation. Therefore, we could infer based on that they do also help memory consolidation. So it's not only the fact, as I mentioned earlier, that they increase the information available for encoding and provide meaning associated with that, but it's also that at a very molecular level, we might say that they help the consolidation, so that memory really tends to stay with us, hence why it's so important. And also there's system consolidation, but that one is such an unknown field to everyone. Us in neuroscience, we have so many things that we're talking about without actually being able to very accurately define what they're based on. And system consolidation relates to how memories are consolidated over a period of like 10 to 20 years. So one example to better illustrate that could be a physician, let's say, that learned in who learned in medical school, or these massive books that we surely love. And then 20 years later, without having consulted that material, they're able to recall that because they learned it 20 years ago, and it was systematically consolidated. That's definitely some field to do more research in. Then there's memory storage. And memory storage, I think that's the most interesting things for psychologists and psychotherapists, isn't it? Because that's basically what you're trying to access when you talk to a patient. It's like all their memories and their past experiences and trying to make sense of that. But what I want to say about storage is actually, so people have argued on whether or not this memory storage is infinite or not. because definitely for long-term memory, there doesn't seem to be any limit on it, but for short-term memory has been agreed generally that it's around 7 objects, plus minus 2 standard deviation, and so on. But one important thing to mention about storage, I think, is that it seems to decrease with time. So basically, the older one gets, the more difficult it is to access it, and that might be due to loss of neurons or everything. So what I think would be a cool idea in this context to make more sense of this particular memory process would be maybe to look at people who are taking psychedelics while they're young and see whether, compared to controls that wouldn't take psychedelics while they're young, and see whether in the future that's going to increase their memory storage or not. And I think that would be a really interesting research avenue, but again, there are no studies on this. So it's just inferences based on the currently available data and anecdotes. Then we have retrieval. And for retrieval, there are 2 main theories around this. So the first one states that the memory retrieval process has 2 states, and one of them is literally like fishing in your memory for the right facts, and then making a comparison between, is that correct? So did I recall the correct material that I called out? Which is more, if you ask me, more of like how computers work rather than humans. But there's also another theory, which is called the encoding specificity memory retrieval theory. And that basically states that memory retrieval is very dependent on the environment. And as an example, we could take depression, for example, where a patient who is depressed on a daily basis is more likely to remember depressive events or really, really sad, tragic stories from their childhood, and that basically led to their depression or so on. But this could also be applied to psychedelics. And I think that here, for example, it's really easy to talk about psychedelics being a way of increasing this memory retrieval. So during an experience, the memory retrieval might be increased, and they might be very useful for accessing that storage, as I just said. But it's also very interesting because based on some reports, like the same memories and the same experiences can only be accessed in the same state. So that's really something important to take into account. And finally, the Reconsolidation framework. So basically, this framework states that by reactivating a memory, so basically by recalling something, there is novel protein synthesis going on. And this novel protein synthesis and all these molecular changes that are happening would allow us to overwrite that memory with novel experiences and novel meaning, because the memory becomes liable by just reactivating it. And that's also how false memories could be involved. And I'm pretty sure that everyone heard of the stories where people have witnessed an accident and afterwards they were told by friends or so what happened, and then they seem like they can't really remember their own experience of the same thing because there are people around them who have had different opinions and basically modulated their memories. So I think that this memory reconsolidation framework is really what could be applied with psychedelics, especially in psychotherapy sessions or in like overriding the old memories with novel meanings. And this could be really useful in disorders such as post-traumatic stress disorder, depression, and addiction. And for here, I did get a question the other day on how is addiction related to memory. I'm just going to briefly explain that. If it's not very straightforward already, and that's because we could take something as simple as smoking addiction. Let's take smoking addiction. Let's just take this person, they don't have any cigarettes on them, anything, they're out with their friends in front of this building, let's say, and then they see someone smoking, so that's called cued retrieval, the smoking being the cue, and the fact that they're addicted to it is the problem. And because they see that, they do crave smoking. And I'm just going to say how to apply the Reconsolidation framework for disrupting that. For example, how I used to do it before back at Birmingham. It's basically by trying to reactivate this and at that moment try to give a drug that's going to make you forget what just happened. And while I was doing that with ketamine derivatives, that would be easily replaced with psychedelics where a novel meaning could be associated with the very act of smoking. So let's just hypothetically state that in this therapy session, a person sees this pack of cigarettes, and instead of feeling that they crave that, they might associate novel meanings of their own with that particular moment. Of course, there are some criticisms to this work generally, both with psychedelics and with memory. And one of the first ones would be ecological validity. And that basically states how well can we scientists replicate what happens in daily life in one of our very controlled experiments. And whether that controlled experiment was structured in such a way it can actually represent real insight into how things are functioning. And you could arguably say that with psychedelics, they do possess this quality of giving meaning to the experience in itself, and could potentially make even a scientific environment mean something and facilitate all the studies. But with memory in itself, with studies based on memory, the majority of them have actually been based on very meaningless tasks, like try to memorize this list of objects and then try to recall them and so on. So I think as future directions for us scientists, we should look for trying to replicate real life better, but that's obviously quite difficult. And another criticism that I would like to bring forward in regards to both of these types of works is priming, and that's more to do with psychedelics really, because with so much information widely available, for people who have, for example, never tried psychedelics before, it's very easy to read everything that's on AeroRead or online and then take your first psychedelic and in a study setting, legally, in a scanner. And then basically try to infer, we will have an experience that's very biased towards what you've read before, potentially, or not. So I think that's really something that we should control for more in these studies. But basically what my take-home message from this presentation would be for you is that psychedelics could definitely be used as memory modulators by using this Reconsolidation framework, for example, and their applications include depression, addiction, and post-traumatic stress disorder, but maybe others as well. And if you have suggestions, come to me later, we can talk about more. And yeah, I just want to thank, really, these are my best friends. It's like, because they encouraged me to like put this forward and so on. I want to thank my lab for being absolutely amazing, and also the organizers of this conference, because it's truly one of the best psychedelics conferences I've been to, and like so much work has gone into this. So like, congratulations, guys. Yeah, thank you. That's my talk. If you want questions. Also, I'm really sorry, but I'm really ill. I hope you could tell. So, like, if it wasn't as fun as I probably advertised it, that's why.
I think that went great, Maria. Thank you very much. Any questions about memory?
Sure.
Here we go.
This one. Okay.
Thank you for your talk. In our day-to-day consciousness, very emotional events become ingrained in our memory and stay with us until the very end. And from these 2 days, from what has been said, most of the psychedelic experiences for a lot of people are one of the most meaningful and memorable in their life. But I think everyone would agree that a large proportion of what we experience during the psychedelic trip is lost. So the question for me is, do psychedelics acutely disrupt memory formation, and what would be the mechanism for that? It kind of, what would you think and say about that? Thank you.
No worries. So, like, I think I had something that, so here, this thing with the encoding specificity one. So, what I mentioned here, I think I wasn't necessarily the most clear, but with psychedelics, I think it's really important to remember that while they can provide a great insight during the experience, what tends to happen is that this in itself, it's a stimuli, just like the feeling of like the depression that I gave as an example earlier. So you might find more likely that like the memories of the psychedelic experience could only be recalled in another psychedelic experience, really. And that is the paradox that I mentioned earlier, and that I find that is indeed one of the challenges that unfortunately I don't have an answer to yet, but I would really love to. So yeah, it's all to do with encoding specificity.
Thanks for a fascinating talk. And this with reconsolidation is now popular in psychotherapy discussions all over. And some authors claim that in order for reconsolidation to take place, you need a mismatch. That is, the memory, in order to really reconsolidate, the retrieved memory must be in discord with the present information. A mismatched experience is necessary for reconsolidation to take place, some authors state. What's your comment?
So wait, keep the microphone. How would you describe a mismatch experience in exactly what you're talking about?
Well, you retrieve a negative memory, but the context is positive, so then the meaning is different, and then reconsolidation starts.
No, so that's exactly it. So disruption of the reconsolidation of the memory.
Yes.
And basically this disruption of the reconsolidation being followed by re-encoding it differently.
Right.
So yeah.
And that the mismatch is a key for that.
Yes, it is a key that the things that the memory would be overwritten with are different from what it initially meant.
Right.
Yeah.
Okay.
Yeah.
Was there a question here? Any further questions? All right.
Hi. I'm just going to comment a little bit, being doing a fair share of reconsolidation studies myself. So the mismatch is definitely something that we believe must happen because, and we usually it is defined as a prediction error. So you predict something will happen. Your brain is constantly predicting what's going to happen, and if that doesn't happen, then there's a point of updating that memory. So. One of the comments to this would be that possibly when you're in a psychedelic state, your expectations are sort of changed.
Yes.
Because your perception is very different. So possibly it could be an extremely powerful way of shaking loose a lot of memories, enabling them for reconsolidation. But it's a bit. I don't know exactly how that would happen. When you give people SSRIs, you can strengthen their consolidation of memory, so you actually would remember things better. So by sort of flooding the brain with serotonin would possibly give a great memory of the experience, but that doesn't seem to happen. So I don't know. I just wanted to sort of comment on this.
So I think in response to your comment, I think it's really important to mention that this is not something that will happen spontaneously. Like it's definitely not, I'm definitely not talking about a spontaneous reconsolidation process being disrupted by a spontaneous psychedelic experience, like you take psychedelics with your friends in the forest or something like this. I'm talking about actually working on creating that type of environment. So that could, I think, in my opinion, only be achieved through therapy. Yeah.
Okay, thank you. So memory consolidation is dependent on the NMDA receptor.
Yes.
And so my question is, ketamine and other NMDA antagonists,
yeah.
do they have a role in memory not being formed?
So I did not look into ketamine much, but into ketamine much in this particular framework in humans. But what I have done is that back in Birmingham in the old days, most of my experiments were actually using a ketamine derivative, which is called MK801 for disrupting this reconsolidation of memories. And what happened with that is that it completely managed to erase fear memories. So actually, I think you make a really interesting point. What would happen if someone tries to disrupt this consolidation in like a human experiment? Like they take ketamine straight after they reactivate a specific memory. But I'm pretty sure that has not been done yet. And people are possibly very reluctant to doing that due to ethical considerations, which are obviously understandable. But yeah, it would definitely be something interesting to explore.
But in the when you're under the influence, do you make less memories?
That I wouldn't know. So, yeah.
Well, thank you very much.
Okay.
Any further questions? All right. Let's give another big hand to Maria.
Okay.
Balaet.